Take a second and think about light. Color, vision, and maybe a little bit of physics probably come to mind. But what about selective breeding of mice, brain cells, and a protein that's typically found in a type of microscopic algae? Maybe not? Okay, let's start from the beginning. This is your brain, the paramount organ in your nervous system. Working like a circuit, it sends signals throughout your body using cells called neurons. Here's a close-up. When a large enough signal caused by a stimulus reaches a neuron, it fires, taking the charge and relaying information from neuron to neuron, causing a chain reaction of electricity and chemistry across your head and throughout your body. Let's zoom in. When a neuron fires, the concentration of charged sodium and potassium ions in and around the cell switch as channels in the cell membrane corresponding to their molecule open and close. With the movement of these particles, the voltage of the cell begins to grow, and more protein-controlled channels open to quicken the process. As this continues throughout a nerve, neurotransmitters, or chemicals created and received by neurons, spread the message throughout the brain at the synapses, or connections between separate neurons. But when it comes to optogenetics, this neurological reaction isn't caused by neurotransmitters. It's caused by light receptor proteins, such as these, which do the same job as other protein channels, but start their reaction from inside the neuron and don't need neurotransmitters to work. These proteins, called rhodopsins, can be found in algae and were first introduced into the neurons of rodents through a virus. In the case of optogenetics, the neurons carry the protein genetically once implanted, allowing selective breeding to pass it on to specific strands of cells in the next generation. Due to this, optogenetics allows for the selective stimulation of the neurons that carry the protein, rather than a typical full reaction from nerve to nerve. Just imagine a cell as a room. Using optogenetics to send a signal is like turning on a flashlight rather than opening the blinds to let sunlight in. Not only is there more control of where the signal goes, but the reaction is also able to directly take place within the cells, rather than go through an entrance, such as a glass window or a series of neurons across the brain. Meanwhile, in the subjects, the light can have various results, depending on the purpose and location of the affected cells in the brain. It's just like the flashlight, it only gives a view of the portion of the room it shines light on. But even if optogenetics works, this raises the question of why and how. Well, even though the idea of optogenetics is relatively new, with both its discovery and first research occurring in the early 2000s, the possibilities of research are broad and wide. As a new way to analyze the brain, it can be used for neurological mapping, allowing us to understand why and how some specific neurons work. This has given us insight on behavioral patterns and the location of specific reactions in the brain. Along with this, gene therapy, vision recovery, and the treatment of disease, such as Parkinson's and epilepsy, have changed due to optogenetics and the mapping it has provided. Now, when you think about light again, what comes to mind? Whatever it is, hopefully optogenetics becomes a part of it.